Hello, my name is Shanika Jackson. I am a senior at School Without Walls in Washington, D.C., and the title of my project is Genetic Variation versus Environmental Contaminants in Striped Bass in the Anacostia River. My project dealt with, with my project, I attempted to analyze single nucleotide differences in 18 fish samples, but to see if a class of chemicals called endocrine disrupting chemicals were mutating a specific gene called aromatase B. My thesis is excessive runoff water that flows into the Anacostia River containing pesticides, herbicides, toxins, and trash can have detrimental effects on the river's health. And this directly translates into my hypothesis, which is that there is a correlation between environmental contaminants, genetic variation, and striped bass. So this picture here is a picture of a catfish, and it's not a striped bass, but catfish are one of the types of fish that are found in the Anacostia River. And this pinkish thing that you see at the tip of its face is a tumor. And these tumors are caused by environmental stresses, and specifically for the Anacostia, the poor water quality that the Anacostia has experienced. The Anacostia River is nicknamed the Forgotten River because it's become so polluted, and the EPA has said it's the second most endangered waterway, or in this case, endangered means polluted waterway in the nation. So my research that I did found that runoff water was responsible for 75 to 90% of the pollution in the Anacostia River. And runoff water is any water that is on the streets that has not been treated by the wastewater treatment plant that we have here, and that carries bacteria, pesticides, herbicides, physical trash, fecal matter, anything really that you could find on the streets, including trace metals, gas exhaust, leaks, anything honestly that you would think that you would find on a street in a city, especially a city as congested as DC. And so this is a really big problem because 2 to 3 billion gallons of this runoff water is dumped directly into the Anacostia River without any type of treatment, and this affects not only the water quality but the organisms in the river itself. So my experiment was done at Georgetown University with my mentor, Dr. Matthew Hamilton, who is not only a professor at Georgetown, but also runs a fish lab there. And I was able to use fish samples, 18 striped bass fish samples, and I was able to amplify aromatase B in these samples using polymerase chain reaction, also known as PCR, and primer design. So the gene aromatase B is a gene, or really an enzyme, that converts androgens into estrogens. And there are two types of aromatase A and B, and the one that I use, which is aromatase B, is found in the hypothalamus. In the hypothalamus, and is prevalent throughout an entire fish's life, which is the reason why I used it. So the PCR tool, which the diagram is right here of how it works, is used to amplify a specific region of the DNA, in my case the aromatase B, and is used to amplify it into millions of copies. And in my case, I was trying to get enough of the DNA to sequence the DNA in order to look at these single nucleotide differences, but it was very difficult for me to do so because these primers that you see are very crucial to your amplification being successful or not. And these are my primer designs. Primers are really important because they're kind of like tags, like small, really honestly tags that go into the DNA and look at the specific region that you're looking for. These were really hard to do because striped bass has not been sequenced before in any type of way, so I didn't have anything to go off of. So these are my gel electrophoreses that I ran. I ran 3, but the third one had completely nothing on it. The first one, as you can see, is really unsuccessful. I only was able to amplify one product, and it was an ambiguous product. And the second one, I was able to amplify 3 out of the 12 that I did, which was exciting, but it was a mixed result. So as you can see, I had inconclusive results, but if I had a chance to do this over, I would look at a broader range of genes and I would look at more types of fish. And if this was successful and I found a systematic way of looking at differences in the nucleotides, I would want this to be a model for other urban waterways to assess poor water quality. Thank you so much. I hope that this was as exciting as it was for you, as it was for me. Thank you for listening.